where we started this project. So we'll talk about a little bit where we started, where we're at now, and where we're going to go in the future. So let me just start sharing my slides here. Get right into it. Okay, you should see my presentation on the screen. Oop, nope, just make that full screen. Okay, it's still, okay, there we go. So it should be full screen, correct? Yes, okay. Let me just get my laser pointer going. Okay, good. So I'm going to tell you about the research we've been working on for the past 5 years on Amitraz resistance in Varroa. So when we think about all the things that affect honeybees, we typically think of, the 4 Ps, right? Pesticides, parasites, pathogens, and poor nutrition. That's a nice, easy mnemonic, and that's true for the most part. Those things are critically important for honeybee colony health. But we also need to include management in there. So the way that we keep our bees can induce some stress, especially when we're looking at the large-scale commercial beekeepers who move bees across the country year after year and raise them up in artificial conditions and locations. So that's something that we have to consider too. Just the way that we keep our bees also induces some stress. But recently, some of the research that's been coming out about long-term impacts of climate change have really shown that that is what's the one factor to rule them all. So even we did some research where we controlled for all these other factors and investigated them. We went into the fall with very low Varroa, very low parasites, pathogens, and pesticides. So everything was looking good, but climate really really impacted those colonies because they were nutritionally deprived because of a drought and the high temperatures. The bees went into winter very lean and unhealthy. So yeah, we need to consider that as a factor as well. So some of these things are under our control, but some are not. Okay, but what I want to point out here is on this picture is that these things all seem to be equally representative of having impacts on honeybees. But a lot of the research we've been doing recently, and this is as a honeybee research community, is showing that Varroa are really leading the cause of the factors that affect honeybees because they are so consistently found across our colonies here in the country, no matter where you're at. Every beekeeper has Varroa mites, and it's not because you're a bad beekeeper, it's just because you have bees. So we need to consider that as an issue. So you wouldn't tell your dog to just, that has fleas, to figure it out and get rid of them. No, you would do a veterinary intervention. Same thing we do here with Varroa. They are necessary to control by any means, not any means necessary, but by appropriate means that result in healthy colonies. And so this is, don't just take my word for it, but this is kind of some of my favorite papers that have been coming out that really show that the scientific community has demonstrated quite thoroughly and convincingly that Varroa mites are very important for honeybee colony health. And I love this one up here in the upper left. You can read this one, just literally it says, Varroa destructor is the main culprit for the death of reproduced populations of overwintered honeybee colonies in Ontario, Canada. Comes out, says it right in the title of the paper that Varroa mites are really bad. And you can see all these other papers up here show that Varroa really do damage on honeybees. But also, too, beekeepers know this. So this is what used to be the Bee Informed Partnership Survey. This is now taken over by Jeff Williams at Auburn. But when we would ask beekeepers, whether you're backyard beekeepers with a few colonies, sideliners with between 50 to 500, then large-scale commercial beekeepers with more than 500 colonies, you notice here, when we ask them, why did your bees die? So we have Varroa, queen issues, starvation, etc. on the x-axis here. You can see here, especially with sideliner and commercial beekeepers, most of them are saying Varroa mites are what killed my bees. So beekeepers are very aware of this. So this is something that I think actually represents a change in attitudes towards Varroa, because 5 years ago, Varroa were about the #3 cause of colony losses. So we've seen this shift in what beekeepers perceive is affecting their honeybees. Oh, and just a quick note here too, is that I want this to be kind of a conversation. So I'm going to be talking with my slides, but if you have a question, we could relay it in the chat and I'll try to answer it in real time. So yeah, if you have any questions as we're going through this, just shout them out, because it's better that way if we have some participation. Well, we say that Varroa are a devastating honeybee parasite. So you can see this bee here with this gigantic, relatively gigantic Varroa mite on its abdomen here. And that's pretty big relative to the size of the honeybee. That would be like if you had a mite the size of a dinner plate on your abdomen. So it's pretty large relative to the size of the honeybee. And you notice that the Varroa mite is on the abdomen of the honeybee. And that's not a mistake. That's not just a good photo here, but it's there for a reason, because that's where they're feeding. Yes, these slides will be available after the meeting. And so if we look at where the mites are feeding, they're feeding on the abdomen. So this is work done by Sam Ramsey. He's at Colorado University right now. And you can see here on the abdomen, these dots here show the areas where Varroa mites were most frequently observed feeding. And you can see they're on this part of the abdomen of the honeybee. Well, underneath that part of the abdomen is this tissue called the fat body. And fat body kind of works analogous to our liver. It produces hormones and peptides and proteins and you name it, the fat body makes it, okay? It's really that versatile. It also has really big important implications in detoxification of pesticides, hint, hint, something that we've been looking at. But it also manufactures this protein called vitellogenin. Vitellogenin is an egg protein that, much like the fat body, does the myriad of things in the honeybee. It's responsible for their transition of workers from nurses to foragers, and also to longevity of overwintered bees. And we know it feeds on fat body because when we put a stain in the fat body and then see where that stain goes in the Varroa mites, the fat body stain, this red stain here, turns up in the Varroa mites that are feeding on the adults. So we know when Varroa are feeding on adults, they're feeding on fat body. However, when the Varroa are feeding on the pupae, they're actually feeding on the bee's blood, which we call hemolymph in the entomological world. And it looks a little bit different. So if we take these mites that were feeding on the pupae, where they're reproducing and growing up, and you can see here on this picture, where they're unstained, they just have this kind of morphology. But when we look at what they've been feeding on, you see how they're only lining up a little bit of red for the fat body. But when we stain them for hemolymph feeding, you notice here they light up much, much brighter. So when they're in the pupae, they're feeding on the bee blood, and when they're on the adults, they're feeding on the fat body. So a little bit different feeding ecology depending on life stage. But I want to point out something really important here is look at this citation. This paper came out earlier this year. So there's still so much fundamental biology that we don't know about Varroa mites that really is going to be really important for understanding this pest biology and how we can control better. So we're still making very fundamental findings on Varroa mites even this year. So much more research on Varroa. And so not only do Varroa feed on the fat body and blood of the bees that could cause damage, but they also transmit viruses. So just like mosquitoes transmit malaria and yellow fever, these Varroa mites transmit the devastating honeybee viruses. So on the left, you could see deformed wing virus typified by bees with these deformed or crumpled or twisted wings. This is what's critical about this virus, though, is that you don't see these crumpled wings unless those virus levels are very, very, very high. If you see bees with deformed wings in your colony, you know you have very high Varroa mites. We know that those two things are limited. More Varroa are related. More Varroa, more deformed wing virus. Conversely, they also transmit chronic bee paralysis virus. Now, you can see them are indicated by these bees that have this kind of shiny, oily, hairless appearance. You know, compare the ones on the right to the ones on the left in terms of hairs. They also move a little bit differently once you see them. And this disease is not, it's vectored a little bit by the Varroa mites, but that feeding, that damage, that stress that Varroa induce make these bees more susceptible to this virus as well. So when we're thinking about what Varroa are doing to honeybees in terms of damage, we have to think not only just about the Varroa mites, we also need to consider the viruses that they transmit. What's interesting about the viruses is the more we look at them, the more we find. So there's still, again, much more research to do. Just you could do a whole career on the viruses transmitted by Varroa. So when we're looking in our colonies, what do Varroa mites look like? Hopefully you never see this. So when you start to see Varroa mites on the adult bees, like in this picture here, this is very, very bad. You can see on this one here, we have 1, 2, 3 mites on the adults. 1, 2, 3. 1, 2 here, 1, 2. So this is bad. If you could see them on the adults, that's a very high infestation. Typically, you would see this late in the season when you start to lose brood. You can see here there's no brood. So all those mites move up onto the adults. So this is something you don't want to see. You may also see them invading the prepupae. So as these honeybee larvae transition to pupa, you can see here in these two cells, we have these foundress Varroa mites invading the prepupae. They'll get sealed up, and that's where they reproduce. Another place we see a lot of Varroa mites are in that drone brood in between your boxes. So if you have any of that, let's say have the boxes spaced a little bit too far apart, they start to build out that drone comb, or you have a medium and a deep box, you might get some of this drone comb, and you can see all those little specks here are Varroa mites. Again, this is extremely high infestation levels. This is kind of a worst case scenario. This is definitely what you do not want to see. But there are ways that we can measure them before they become too much of a problem. And so some of the ways that we could do that are with screen bottom boards. So this is a screen bottom board. You would put your boxes on top of this as you build your colony. And then you could slide this tray here, usually coated with petroleum jelly, to measure how many mites fall off of your bees. The more mites falling off, the higher infestation you probably have. It's a good way to passively sample Varroa instead of having to sacrifice any bees or damage any comb. But this is a way to passively measure, okay, do I have a Varroa problem or not? One way you could speed that process up is by using powdered sugar. What this does is it causes the mites to lose their grip on the honeybees, and also it induces the bees to start grooming each other to knock the mites off. You could typically get a good amount of Varroa to drop off the bees within about 2 hours after you hit them with powdered sugar. So if you want to speed up that mite drop, that's a good way to potentially do that. But the way that we typically count mites at the lab for research purposes is we do these jar tests, and we'll talk about how we remove the Varroa from them. So what we'll do first thing we always do is we always make sure we don't sample the queen. We'll find her, put her in a cage, put her off to the side. Because what we don't want to do is shake that queen into this bucket as part of our sample. So please take caution to not sample the queen. So we'll usually get bees off of brood comb. So wherever we have sealed brood, we just put over this bucket, shake the bees off really quickly, then use a half cup size scoop of bees, typically about 300 bees, to do our jar tests. So when we put them in the jars, there's a couple ways we could get the mites off. The first way is our old friend powdered sugar, I just mentioned before. This works pretty good if you don't have a lot of humidity, but in Louisiana, we can't do this that well because powdered sugar turns to a rock in about 5 minutes as soon as you get in the field. But if you have a place where you have low humidity, you could put the bees in the jar. You can see this jar is just a mason jar with a screen lid. Add about a tablespoon or 2 of confectioner's sugar. There's not really too much difference between them. Roll them around a little bit. You don't want to shake them, but just roll them around for about 30 seconds to a minute. And then you could shake the mites through the screen onto a tray. You can see a couple of these dots here. You could then use a squirt bottle to dissolve the sugar and measure your Varroa mites. This method's good if you don't want to kill your bees. So if you want to return back to the colony alive, that's a good way to do it. But what we do at the lab is we use either detergent, basically Dawn dish detergent, or alcohol, 90% ethanol. We put them in these jars, shake them up for a while, and then when we pour the bees out through the screen, we could get the Varroa mites in this tray down here in the bottom. This does 2 things. It allows us to count the number of Varroa, so we can get that count, but we could also standardize it by the number of bees in the sample. So typically, and in this presentation, you'll see this expressed as Varroa per 100 bees. So that way, if we take big samples or small samples, we could standardize by the number of bees in the sample, which is important for doing an apples to apples comparison. So I just want to show this because I think it's really incredibly important for us to start thinking about Varroa inspections now, because the sooner you detect Varroa and start treating for it, the less of an issue it'll be later in the year. So make sure everyone's counting Varroa mites, please. So you counted Varroa, you have infestation. What do? Typically, what most beekeepers do is we turn to miticides. One, because when they're used according to label and you have miticides susceptible to Varroa, these materials tend to work well at reducing Varroa populations. However, some of these materials have better efficacy than others. Now, up here, we have some products that have these active ingredients. So Apivar is the product that contains Amitraz. Checkmate is the product that contains coumaphos. These are the active ingredients: coumaphos, formic acid, fluvalinate, hospate acids, so on and so forth. Now, when you're looking at this slide, it might look, oh, we have plenty of row control options, so we don't need to worry too much about this. Just pick one and they should all work. Well, I wish that was the case. People don't use products containing coumaphos or taufluvalinate very much because resistance to those products have developed a long time ago, and they're practically non-functional in most commercial beekeeping operations anymore. So very few people use those materials. Products containing formic acid, oxalic acid, or hops beta acids have some unvariable efficacy, mostly because they are most effective when they are used without brood. So typically in the wintertime, which I don't think is too long out in California, in Baton Rouge, we barely have any winter time where we have a strong brood break. So these materials have limited efficacy in these environments where we have a continuous brood presence. So not always able to be used. And also, too, they require extensive, just because they're organic acids doesn't mean they're safe. You look on that label of oxalic acid, you'll need to wear the full face mask respirator if you're going to use it as a vapor. So pay attention. And they also have temperature requirements like formic acid. You don't want to put that stuff in your colonies if temperatures, I think, over 85 degrees as per the label, because it'll damage the brood and run off the queen. So again, limited use for those materials. So what most beekeepers use are products that contain Amitraz and thymol. And so what has happened is that people got on the track of just using Amitraz products all day, every day. The only thing they do for miticides, for Varroa control. They're not using integrated pest management. They're not using any of the other tricks that we'll talk about. It's just always been Amitraz. Well, when you use the same thing over and over again, these mites and other insects in general, they develop resistance. This is just the natural outcome of a selective force on a population. And that's something I studied during my PhD. So this was kind of a very a topic that was near and dear to my heart. So once we started hearing beekeepers saying that Amitraz isn't working like it's used to, and they had to increase the dosing and the frequency, I knew we were dealing with resistance. So I'm going to talk to you a little bit more about the project we've been working on for the last 5 years. So just give you a brief overview is that we'll talk about the definition of resistance and what the prevalence of it is. We'll look at how we could use this test to predict if Amitraz-based treatments will work or not. We'll look at the genetics of Amitraz resistance. This is a whole new emerging field. Like I said, so much more work to do on Varroa mites. And what's most really important here is strategies to overcome resistance. This isn't the end of the game here. We just have to think differently here and use different materials and different strategies to overcome resistance. So how do we define Amitraz resistance? It's very simple. So what we do is we take a scoop of about 300 bees, like I said, about a half cup of bees, and we put them into this very sophisticated custom built, it's a deli soup container. It costs about $1. I buy these by the case from my restaurant supply. So we put those bees into that deli container. And then you can see here on the top, there's a square of Apivar. We just cut the strips of Apivar that you would get. We just cut them as long as they are wide to make this little square. And then we hot glue it to the bottom here. When the bees are in that container, we put this screened lid on the bottom. This is a screen lid, about 1/4-inch shot cloth. And then we suspended it over this tray that's coated with petroleum jelly. So that way when the Varroa mites fall off of the bees, they get stuck on the tray. When those Varroa mites fall off, that means that they are susceptible to Apivar or Amitraz. So when they fall on the tray, we record the number of Varroa that fall on the tray, those susceptible Varroa. After 3 hours, and I'll explain where we get 3 hours a little bit later, We then wash the mites off of these bees with just some soapy water. It's pretty easy to do. And then we record how many mites are still on the bees after that 3-hour interval, because by definition, those are the resistant ones. So we count the number of susceptible Varroa on the tray, plus the number of resistant Varroa in the washes to get our total Varroa in the sample. And the way we determine Amitraz resistance is we just take the number of Varroa in the wash divided by the total Varroa in the sample. So let's say we found 5 in the wash, but 10 total Varroa. 5 divided by 10, 50%. If we had 10 Varroa in the wash and 10 in the sample, 10 divided by 10 is 100% resistance. So it's a very simple, easy way to do this. I know there are more complicated ways to do this, but this is just a simple, easy way that make it available to use to everybody, even non-scientists. Hint, hint, beekeepers. So this is a test that you could do in your bees. So why do we use 3 hours? Well, we use time as a variable because, like I said before, we use APIVAR, and APIVAR has a 3.3% formulation level. So the concentration is held constant. Now we have to use time as our variable. And so what we did is we looked on those trays and counted what time do all the Varroa fall off. And we found that at 3 hours, 100% of the Varroa fall off. So 180 minutes, 3 hours. And we know that all those mites are falling off due to APIVAR because when we have no square, no treatment, we get very little control drop, maybe 2-3% control mites fall off just on their own. So most of those mites are falling, an extremely high majority of those mites are falling due to APIVAR. So 3 hours, if they all fall off, they're susceptible. Anything remaining on them is resistant. So that's how we came up with that 3 hours. I'll skip this slide. All right, so we've done this test all across the country. So these dots here indicate the locations where we did the resistance testing. So you can see here, this takes us a lot on the road. This year, I'll be going from Michigan through Minnesota, and then out through the Dakotas, Montana, Oregon, and Washington. So my girlfriend's not too happy. I'll be spending 2 months on the road, but hey, we gotta do we gotta go where the bees are. All right, so you can see here quite a broad geographic scale of sampling. Well, let's see how this project developed over the years. So in 2019, we did most of our sampling in New York and in North Dakota. So we worked with 7 beekeepers, 11 apiaries, and we only sampled 83 colonies, but we were able to find that there was Amitraz resistance out there in the sample. Well, in 2020, we couldn't go anywhere because of COVID. So what we did is we started shipping these kits all across the country for people to do the testing remotely. This test is very simple. We have the protocols, there's a video and everything like that. And beekeepers get into this. So this is something that we can make available. So started working with the Apiary Inspectors of America. And again, we doubled the number of beekeepers, apiaries, and colonies in 2020, despite not being able to leave Baton Rouge. So this was actually one of the ways that we've advanced this project is by doing remote sampling. Well, in 2021, we were back on the road again, but we had some help with the Honeybee Health Coalition and Jeff Williams at Auburn. Again, 27 beekeepers, 45 apiaries, 426 colonies. So again, doubling our sampling effort because this project started growing very organically. 2022, same story, more beekeepers, more apiaries, more colonies. This is when we spent a month on the road with the Bee Informed Partnership working with commercial beekeepers to collect 700 samples. That's a lot of work. So, but fun work that we really enjoy because it's really important. And then last year, we mostly just focused in on Florida. So I collaborated with Amy and Jamie and Cameron from the Florida lab to collect a bunch of samples across Florida. So we've been having pretty good representative sampling across the country here. Well, let's look at some of the good news that we found from this study. This isn't all doom and gloom. We actually found a lot of colonies with no Varroa mites. So can't have Amitraz resistant mites if you don't have any mites. So about 22% of all the samples we collected, this 1,655 samples that we collected over the course of these 5 years, 22% had no Varroa. So that's a good thing for 2 reasons. One, it shows that there are ways that beekeepers can keep Varroa levels at 0. 0 is a good amount of Varroa. So yes, this can happen. And even in large commercial beekeeping operations, people running tens of thousands of colonies. So this can be done at scale. So it could also be done at a small scale in terms of backyard beekeepers too. Other thing that we found that was pretty good news is we found 70% of the samples had less than 10 total Varroa. Why do we have 10 total Varroa? What's the deal there? Two things. One is that 10 Varroa per 100 in the sample translates to about this 3 Varroa per 100 bee threshold. I'm sure you've all heard about that as your economic damage or your treatment threshold for Varroa mites. So that's why we consider this below the action threshold. Second is from a resistance standpoint, is that makes sure that we have enough Varroa to be confident in our results. Because if we have 2 Varroa in the sample and 1 fell in the tray, 1 still on the bees, is that really 50% resistance? I don't know. So well, actually I do know because we've done the statistics and we did the genetics, and yes, it is not truly 50% resistance. So because of that, we know that there is some slop in the bioassay, so we can overcome that by sampling only those ones with sufficient Varroa. So again, 70% of the colonies we sampled didn't even have enough Varroa for us to confidently assess resistance. So these two things are actually really good news, if you ask me. All right, well, let's start talking a little bit about the elephant in the room. So there's still 491 colonies with 10 or more Varroa. So this allows us to confidently assess resistance. Again, about 26% of the samples had 0 resistance. So even though they had a lot of Varroa, all of them were susceptible to Amitraz. So this is still good that 26% of the colonies has very, very high levels of susceptibility. If we treat those colonies with Amitraz, it should control them. So this is good news too. Well, then about 36% of the colonies had what I'm calling intermediate levels of resistance, 1 to 29%. This is intermediate. Amitraz may or may not work. We're not exactly sure in this case. But what we're calling high levels of resistance is whenever they have 30% or more. These are high levels of resistance. This is resistant colonies. About 38% of the colonies out of those 491 were at levels of resistance that'll lead to treatment failure at the colony level. Well, now you're probably asking, well, how do we come up with that 30% threshold? It's based on this graph. So on this graph here on the x-axis, this is just a random code that I generate for beekeepers and apiaries. If you could figure out this code, you should probably be working for the CIA because this is all random numbers I created in my spreadsheets. So with that in mind, anonymity is baked into this, so no one will ever know. You're the only person who will know what your Varroa resistance levels are. So yes, again, x-axis, these are just the different apiaries within beekeeping operations. Then on this axis here, we have the Amitraz resistance levels. So 0 is low resistance, 100% is high resistance. And again, you can see here, we have some colonies that have no resistance, that 22%. Then we have the gray bars here, these are the intermediate ones. Then all the ones here in red are above that 30% threshold. But you notice these ones here with the candy cane stripes on them? Those are the Amitraz resistance levels that we found in those apiaries during an active Amitraz treatment. So therefore, we know that level of resistance is associated with treatment failure at the colony level. Okay. So that's how we came with that 30% threshold, because when we see the resistance levels during an Amitraz treatment, that tells us that over 30% should lead to failure. Okay. So that was an observation that we saw. And what we've been seeing over the past 5 years is this increasing trend of resistance. So this is 2019 when we started the project, and 2023 this past year. And you can see here, there's this line here shows that there's this upward trend, whereas in the first year, 10% of our colonies had a resistance, and now we're dealing with about 40% of our colonies having resistance. That will result in treatment failure at the colony level. So this is really important to see that there is this increasing trend over time. I don't know if this is a real bona fide trend, or are we just sampling those beekeepers who have problems? So we are doing a lot more sentinel maintenance monitoring going forward, just to make sure that this is not a biased sampling. But I think that this might be true. All right, so how can we use the test for making you a better beekeeper? So how does the resistance level predict treatment success or failure? And we did this 2 years ago, where in August, We did the resistance tests from 101 colonies from 16 apiaries across 4 states in August. So when we did the test, we get our resistance levels and the number of Varroa in the samples. After we did those tests, we told the beekeepers to apply Amitraz right after we're done. So we're done. We went out there on Tuesday, Wednesday, they're putting out the Amitraz. And then 6 weeks later, we measured the Varroa populations again. The thinking here is if the colony is scored as resistance, we would expect mite levels to increase. If the mites were susceptible in the test, we would expect those populations to go down. Well, what does the data say? In those colonies that we classified as resistant, having 30% or more Amitraz resistance, the Varroa populations in those colonies increased, consistent with the idea that that 30% threshold is going to result in treatment failure at the colony level, as shown by the increased mite populations after Amitraz treatment. Colonies that were scored as susceptible below that 30% threshold, those Varroa populations in those colonies decreased just exactly as we expected based on the resistance test. So therefore, this is a very strong indicator if Amitraz is going to work in your colonies or not. So a very important tool. Now, this is really important because if you're a large-scale commercial beekeeper and you're treating 20,000 colonies and it doesn't work, you're in for a lot of problems. So that's a little bit about the testing we can do in these cages, but are there genetic markers that we can use instead? Because it's a lot easier to do genetic studies nowadays. It's actually very cheap and inexpensive. But what I like about doing genetic testing for resistance is that people could just send us the Varroa mites. I don't need to get on the road. I don't need to go out to apiaries. I could sit in my lab and let the packages come in, and I have great technicians who can run all the samples and determine if they have resistance or not because of the work we've been doing on this. So we've invested in a lot of research in this area, all done with Ariane Avalos. Ariane's a crazy geneticist. So when I did my training on genomics 25 years ago, it was state of the art back 25 years ago. But I'll tell you what, it's been a blinding how fast the worlds of genomics change, because the training I got 25 years ago, which was supposed to be state of the art, cutting edge, is so antiquated and obsolete. It's like a Model T versus a Ferrari compared to what Ariane could do. Ariane's worked with some great people, and he's whenever he talks to me about how he's doing his analysis, it's like whenever you go talk to a mechanic and they're talking about what's wrong with your car, like, yeah, your flux capacitor has gone bad. You need 18.2 gigawatts to get that back up. And you just nod your head because you have to review what they're saying. That's what talking to Ariane's like, but he knows exactly what he's talking about. So it makes it a really good collaboration. With project support from Project Apis M and National Honey Board, we were able to take those Varroa mites that we found were resistant or susceptible in the test, and then we did whole genome sequencing on 432. So this is a very strong, conclusive data set of our samples. So we're very proud of this work. And what we're able to do is do what's called the Genome-Wide Association Survey, or a GWAS. And we're able to find the genes associated with Amitraz resistance. And I'll show those results on the next slide. Not only can we talk about resistance, but we could also talk about population genetics and look at the evolution of resistance in these Varroa mites. So there's a lot of powerful data we can get out of this. And we're always finding new things whenever we ask new questions. Look at the data. All right, so how do we find the gene that is involved with resistance? Okay, so on the x-axis here, we have the genome position. Now imagine every piece of DNA in that Varroa mite was laid out in one long string, where 0 is one end of that string, and above 800,000 here is the other end of that string. So imagine just one long string. And then what Ariane does is he runs this, we're going to call it strength of association test here. So the bigger the peak here, the more of these dots, the stronger the association. That region of the genome is associated with resistance. And you can see here there's a couple little dots that peek out here, there, and otherwhere. But notice here, boom, this big, massive spike. When Ariane saw this, he said, I don't even need to do any other studies because that's such a strong signal. 10 out of 10, that's the gene that's involved with resistance. Well, when we zoomed in on it, we found out that it was this octopamine receptor beta 2 with this Y215H mutation. Okay, again, speaking different languages here. What's interesting about this receptor, the gene that's underneath that peak, this is the receptor for Amitraz. So this is a target site resistance mutation. And it's a specific mutation, this Y215H. This is just nomenclature that says the amino acid in this protein, tyrosine, at position 215 of this string is mutated to a histidine. Okay. You don't really need to know too much about it, but what it shows is that this mutations potentially involve resistance. Because under normal circumstances, in the regular version of the receptor, Amitraz binds the octopamine receptor and it activates the receptor. However, where we have this receptors with this mutation, this Y215H, Amitraz does not bind and does not activate the receptor. Therefore, it becomes insensitive due to this mutation. So now this makes perfect sense because the gene that's underneath that peak with that strong genetic association is exactly what we'd be looking for. So this makes me very, very happy. So now we could use this as a marker, because if you see here on the left-hand side, we have in the test the resistant Varroa that we get from the wash and the susceptible mites that fall onto the tray. If the mites are resistant in the test, meaning they're still on the bees after 3 hours, and we get them off in the wash, there's a 92% chance that they have that mutation. So this is a very strong association of this resistance mutation with the resistance phenotype. So those mites still staying on the bees after exposure to Amitraz. There is a whole other, you might be asking some questions. Well, why are there some, what's going on with the susceptibles? There is a whole other talk I could give on that. But today I just want to show you that, yes, the resistant genetics are definitely associated, highly associated with the resistance phenotype. All right, so now this begs the question, what can we do to get rid of this stuff, right? Fortunately, there is a lot that we know from other systems in resistance management kind of things that we know that there are certain materials to apply. So I'm going to tell you about some of the things that we've been seeing in the field and what we're doing experimentally. But I want to really point out, first off, the importance of good note-taking. People always ask, what's the most important tool in my beekeeping arsenal? A pen. Taking good notes will make you a better beekeeper today. I guarantee it. And this is an example of where good note-taking by a beekeeper really helped us out. So in August of 2020, this beekeeper put on apivar strips to control Varroa populations. And then in October, when the strips were removed, he said, man, there's still a lot of Varroa out here. I don't think this stuff is working. So we were able to get a test done very shortly after on October 6th. And when we did the test, guess what we found? We found out that every colony we sampled had more than 30% resistance. You can see colonies 1 through 10, Amitraz resistance levels all above that red line, that 30% threshold. So therefore, we know that this was the inefficacy here was due to resistance. We also found very high levels of Varroa mites. You can see here in colony number 1, 40% of those bees in the sample had a Varroa mite on them. Now, for reference, 3 mites per 100 bees is that action or the treatment threshold of the damage threshold. So you can see here, extremely high Varroa populations in these colonies. So treatment failure due to resistance leads to high Varroa mites. Okay. So beekeeper says, well, what do I do now that I have Amitraz resistant mites and lots of them? I said, well, as a scientist, I can't make any recommendations because I don't have data. I said, but what you could do is do a treatment, definitely not Amitraz, and just let me know what you find. So the beekeeper agreed, and next day, hit him with oxalic acid vapor. You can see the 7th, 12th, 17th, 22nd of October, then the 6th of November. Hit him hard. Then the next spring, came back with some more oxalic acid vapor. Then in August, again, the following year, we came back and did the resistance test. Lo and behold, what did we find? Well, you can see in 2020, the bars, the solid bars, blue bars shown here, in 2021, only colonies 7 and 8 were able to find any Varroa mites to detect resistance at all, and they're all below that 30% threshold. So resistance went away with the rotation of other miticides. In a similar note, too, that the number of Varroa also went down dramatically. You could see here in 2020, the solid red bars the year before, and after the rotation in 2021, the striped bars here show that Varroa populations were significantly reduced. So this is just a good example of how rotations can work. But when we see this over and over again in other beekeepers who had similar problems, this seems to be one of the silver bullets here is just use something else. Don't use Amitraz if you have resistance. Rotations can work. Now, these are all observations. We are reproducing the heck out of this with another. We did this in the fall of last year. We're reproducing it again this year just to make absolutely sure that those results are true. So we can make a very confident, data-driven recommendation on rotations. But the data so far suggests that rotations work at getting rid of resistant Varroa. Okay, now we're going to talk about another thing that's near and dear to my heart is using Varroa-resistant honeybees. So you don't have to apply miticides. The bees do all the work. So on this figure here, it shows the process of how as honeybees grow up from this egg that's laid by the queen, it develops into a larva. And right before that larva turns into a pupa, the founder's Varroa infests. And then as that honeybee pupa grows and develops, so does the Varroa population in that cell. And then once that bee emerges as an adult, it'll emerge with offspring Varroa. So this is the Varroa life cycle. Well, bees that have this Varroa-sensitive hygienic behavior, or VSH, what happens is that as those Varroa mites invade the cells and begin to reproduce, so laying eggs, you can see that here, here's some offspring, we think we know the chemical cue here, but what happens is the bees have to detect those cells infested with Varroa. So detection is the first step, uncapping is the second step, and then removal is the third step of Varroa-sensitive hygienic behavior. So this is what it looks like in a figure. Let's look at a video. So this bee here detected that this cell is infested. So it's uncapping the cell. And you'll see when she moves away that the cell is they're developing a hole. You see that there. And now we go to the removal phase where they are destroying the pupa. So destroying the pupae interrupts the mite's reproductive cycle, and she is not reproductive. So you can see at the top of the cell, you see they're removing the pupa. Here comes out that female Varroa. She's non-reproductive. She hasn't laid any eggs yet. So this interrupts her ability to reproduce. And you can see when she gets on the bee here. Okay. So this is sufficient enough to keep Varroa populations very low. Well, you might say, how low? 0. Yes, 0 mites in colonies with bees with this Varroa-sensitive hygienic behavior. So VSH is a behavioral trait. It's not a stock, it's a trait. We developed a stock of bees that have VSH. You probably have VSH in your bees out in your colonies at some low level. In our colonies, it's very high level. In your colonies, it's probably low level. This is a hygienic behavior that results in low mite reproduction. It's all about non-reproduction. You could have Varroa in those colonies, but if they're not reproductive, that's the key here, non-reproductive mites. And in colonies that have a good expression of VSH, it confers strong resistance to Varroa. So I think a very important part of beekeeping in the future, because it's literally no work on your part other than putting those bees in your colony. Another thing that we can use to keep Varroa mite levels low is using Propolis, that waxy, resinous, sticky stuff that gets in your frame rests and in between your boxes. Propolis is this mix of plant resins, beeswax, and other products that go all over the colony that are used mostly for structural insulation and controlling airflow and humidity and things like that. But those plant resins have antimicrobial properties that get rid of chalk brood viruses and things like that. Mike Simone-Finstrom, that I work with at my lab, is one of the world's experts on the ways that bees use Propolis. So we started to say, well, if it keeps microbes low, can we maybe use this for Varroa? Because we started seeing this small signal. Excuse me? Oh, was there a question? Okay. So what we did here in this study was on the left, here are our control boxes. So this is what you probably have. These are just what you would get from any beekeeping supply. And you notice here they have this smooth interior textured. The ones on the right here, we call these our propolis boxes or rough boxes. And on the interior, they have these grooves that are cut into them that are, you can see how rough they are. They're about a 1/4 inch wide by a 1/4 inch deep, every 1/2 inch on the inner surface. That textured surface, there's something about it that stimulates the bees to pack that full of propolis. And you can see all those dark spots here are packed in with propolis. So we know that we can get the bees to put more propolis. When we score this visually, we take pictures and have people score it. So this would be on the left, that would be a 0 or a 1. This would probably be a 5 or a 6 because of how much propolis there is. Well, what we found is when we did this study is that it really knocks Varroa levels down. So on the x-axis here, we have the dates we did our observations. And then the bars here, the white bars are the control colonies, ones with the smooth surface, low propolis. And the gray bars are the propolis colonies, the grooved interior, so lots of propolis. And then we have our Varroa infestation levels here on the left. You can see here, we started them off with the same amount of Varroa in June, but then in August, they had half of the Varroa mites in the high propolis boxes. And then in September and October, they had between 5 to 7-fold less Varroa mites without any other treatments besides those propolis boxes. So there's something in that propolis that's keeping Varroa mites low. What it is the $1 million question. It might actually just be miticides because it turns out propolis does contain a number of miticides and agrochemicals and things like that. But we don't think it really affects the honeybees' survival or productivity. But it may offer this benefit to keep Varroa mites low. So this is an experiment that we're reproducing this year with bigger numbers and bigger boxes too. So stay tuned for those results in the next few years. So again, not doing anything else besides just adding boxes that stimulate the bees to do all the work for you. All right, so I wanted to finish up tonight with just by talking about recapping what we did. Is that we've been working on this problem for about the last 5 years now, and it's becoming an issue more and more across the country where beekeepers are changing their miticide application regimes, and reusing these Varroa resistant stocks are becoming more and more necessary in order to keep Varroa levels low. So this is a problem that isn't going to fix itself. We have to actively work on this. So it takes beekeepers from people that have 1 colony in your backyard up to the people that have 100,000 colonies. We all need to be concerned about this Varroa problem. We know that there are tips and tricks we need to do. We need to start using other miticides. Don't just rely on Amitraz. Use other materials as well. Use other materials, use hygienic stocks and things like that, because this isn't the end of the world. There are solutions here, and we will be working on them aggressively over the next 5 years. So Mike Simone finished them in my lab. We have that propolis engagement, but also too, we just got this huge grant with, I think, like 21 of us all across the country to do more Varroa miticide research in light of all these resistance problems. So this is something that we're definitely working on. This is something that you could participate in as well. I could ship you the kits to do your testing. We'll go over the results, and we could recommend a treatment if you should use Amitraz or not. You'll get your Varroa levels too. So this is still an active area of research that I don't think is ever going to stop. So this is my life now. I live in the world of Varroa. So I'm going to turn over the question, turn it over to everybody to ask questions, and yeah, we'll have a little discussion here. Okay, so Nikki asked, can one paint propolis on the inside of hive bodies to get the same result? We haven't tried that. I know that people, you can buy propolis, I mean, on the market, but we don't know if like melting it and painting on the inside will have that same effect. Do bees need to actively forage for it? We really don't know. That's actually a really good question. We won't look into that, because what's interesting about propolis, it has kind of like a terroir to it. Is that the propolis that we get down here in Louisiana is different from the propolis that we get up in the Dakotas and anywhere else, and it's very, very different than that green propolis that we get from Brazil. So I'm sure there has to be some kind of plant chemical in the propolis that we would get here that would be different from anywhere else. That might actually be what may be contributing to the miticide activity of propolis. And in fact, we're working on that with a plant chemist at our USDA lab in Florida to kind of get at that question. So yes, I collect
Hello.
Propolis. Okay,
Uh,
good.
where do you get your VSH bees?
So we raise them at the lab. This has been a program that we've been working on in the lab for about the last, oh boy, 25, 30, close to 25 years. We have what are called the Poll line honeybees, POL. It just stands for pollination line. And this has been an ongoing process because the first round of the Varroa sensitive hygienic bees, they're really, really good at keeping Varroa mites low, but they were not ideal for beekeeping. They tended to have smaller colony sizes. They would get this hyper uh, hygienic behavior where they just remove all the brood, which is not good for beekeeping either. And they didn't tend to make a lot of honey. But what we did is we incorporated genetics from other beekeepers to make this kind of the best of the best. So now this Paul line, this VSH 2.0, if you want to call it that, has high honey production, high survivorship. They're very gentle. They're light colored. So they're basically everything that people like in bees. That's what we put into them. But they also have high levels of Varroa sensitive hygiene. Now, I want to say something that's really important here is to ask questions of the people who might be selling VSH stock. Ever since I've been giving these talks on Amitraz resistance and also to the breeding stuff that we've been doing, seeing a lot of people selling resistant stock out there. It's really important, and I could give a whole other talk on things you should ask about breeding for VSH because you can't just buy a breeder queen and graft off of her and sell VSH queens. It takes rigorous selection, a controlled mating program, and validation, selection, breeding. So I could send a list of questions that you should be asking people selling VSH. Now, I can't make any recommendations, and this is not an endorsement of the performance of any of these bees, but people like Corey Stevens in Missouri, he has been doing breeding the proper way. I grilled him for like 2 hours at my lab, and he knew what he was talking about. Now, that is not an endorsement of his bees. I just want to be absolutely clear about that. But it is that recognizing that he is doing selections the right way. Adam Finkelstein is another one at VP Queens who has got material from us a while ago and has his own breeding program. Again, not making a recommendation, but Adam has been doing the resistance selection in a way that is consistent with what we do at the lab. Part of what we're working on the next 5 years, though, is working with all the queen producers across the country, especially big queen producers, because they realize that there is a shift in the marketplace that people want hygienic stock. So we're going to work with them to teach them how to breed for hygienic stock. I think you're going to start seeing more hygienic stock in the marketplace in the next few years.
What are your numbers for the using the powdered sugar on the bees versus the chemicals?
So is looking at when you say powdered sugar, do you mean how does that compare to like a
The numbers for the insecticides, like the numbers, like if you're using
so the number of mites that would fall off in the jar tests?
Yeah.
So it's pretty comparable. We've done some of those experiments where if you just use powdered sugar, you'll probably get it depends on temperature, not temperature, humidity. If you have low humidity, you can probably get about 95% efficacy compared to the other methods. But when it gets too humid, that powdered sugar clumps up and it loses its ability to knock the mites off, and it makes the bees really sticky and gluey. It's kind of gross. But yeah, it's not 100%, but it's very good, especially where you have less humidity. And it's also good if you just want to kind of get like a good estimate of the number of mites in your colonies. Yeah, you can use powdered sugar, and especially too, it's not lethal. So.
All right, very awesome talk. Learned a lot. So I have two questions. So first one is like, as a precautious like kind of way of doing this, like where we should looking at like as like kind of early infections, like possible area with the for the Varroa mites infection. And my second question is like I like the rotation of the treatment, like kind of just control the resistant. But are you kind of, I guess, let's in a broader way, like, are you worried about like raising some super resistant Varroa mites? Right. That's okay.
So state that first question again. Just want to make sure I
So
understand.
for precautious, kind of like way of doing this, like where should we looking at as the early infection of Varroa mites in the hive? We're looking at, yeah.
So what time of year should you be doing this?
No, like the where is, like, should we looking at the adults? But adults
Oh.
seems to be a like a late stage already, like very serious. But like, should we looking at the brood or like the eggs or
Yeah.
like,
Yeah. Okay. Yeah, I get it now. Yeah. So what we're finding out is that just on any adult bees, that'll give you a good indication whether it's. We just sample them off the brood frames, just that way we're sampling things consistently across colonies. So you could do it on. I would recommend just getting bees off the brood frames. Look at the adults, but also too, when you're looking at the bees in your sample, look at how many drones you have, because there's been some research coming out that shows that Varroa tend to aggregate on the drones. So if you have more drones in your sample, you potentially can have more Varroa mites because the Varroa are more attracted to drones. Whether that's on the adult drones or even in the drone pupae, they'll typically have more Varroa mites in them. You, as a practical beekeeper who's not doing this for research purposes, I think you could get away with just looking at Varroa on the adults. But if you want a really good hardcore measurement of the total, a good estimate of the amount of Varroa in your colonies, the infestation levels, is you could get a microscope and some nice forceps, little tiny fine forceps, and open up those brood cells, those pupae, and find out how many of them have Varroa in them. So that way you can see number of Varroa on the adults, number of Varroa in the brood. Because rule of thumb is that whenever you have capped brood present, there's about twice as many Varroa in the brood than there is on the adults. So just because you don't have too many on the adults, you could have some in the brood. So it depends on the level that you want to get into it. Other things you want to look at, consider too, is time of year, right? So one mite in your bees in October is really, really good. But one mite in your bees in April is not so good, because just the way that the populations grow, they grow exponentially. The longer we can keep that line flat until it goes up later in the year, the better. So one mite early in the year is not as good as one mite later in the year.
Thank you so much.
Yeah. And then the second question, say that second question again.
Yeah, the second question is like, I read about like, rotation the treatment like can end up with some like super resistant bees, super resistant Varroa mites to multiple treatment. Or...
Extremely unlikely, and mostly based on systems that we found in other insects. There's this idea that if you're resistant to one, the probability of being resistant to one material is kind of low. Let's say, just for example, if we look at all the samples we collected, right, the probability of being resistant to Amitraz is, let's say, I'm going to make up a number here, just I'm going to say 25%, right? But the probability of also being resistant to another material, if it's 25%, what's 25 times 25? Like 5%. There's a very low probability that you'll be resistant to both, right? So that's just at the genetic level. So very low probability of that. However, what happens is that unless there's cross resistance to those two things, so if being resistant to material A makes you also resistant to material B, if you have that cross resistance, then yes, you can develop the multiple resistance like that. However, all the materials that we use for Varroa mites, they don't have that kind of cross resistance. So the chance of being resistant to two things is very, very low. Second, what happens is that whenever you have let's say you're resistant to Amitraz, but you hit them with oxalic, right? They're going to die for the most part. There's never been any report of resistance to oxalic or formic acid or hops beta acids or thymol. So it's only our synthetics. So looking at Amitraz, like I talked about today, taufuvalinate, the active ingredient in apistan, and coumaphos, the active ingredient in checkmate. The organic acids, not just here in the United States, but around the world, we haven't found resistance to it. And I think it's because resistance develops when there's a constant selection environment, when there's a constant demand for them to have some sort of resistance. So when you hit them with oxalic, what happens is, let's say the vapor, that selection pressure is only exerted during the time that that vapor is in the colony. When the vapor's gone, that selective force imposed by oxalic acid goes away. So the mites that are under the caps or whatever that evade that treatment, they're still going to contribute susceptible genes to that population. What happens with things like Amitraz is when you're taking a strip in your colony, taking it out, put a new one in, taking it out, putting a new one, providing a constant exposure, it mandates that the Varroa mites develop resistance because they have to come up with a way to survive that constant exposure. And that's what I think we've been doing with Amitraz. So what I'm trying to say is that it's highly unlikely that we'll develop a super Varroa mite. Yeah, I don't think so. Not with Varroa. I could talk all day about that. That's what I did my PhD on, was insecticide resistance management, so I could get into like nitty-gritty details. I love it.
I have a question. Did you ever try brood breaks, kind of like system, isolate the queen and let them create broodless period?
Yeah, broodless period is a big thing in Europe. I'll tell you that.
They keep queen in a cage for 6 months.
Yeah. And it's interesting because, they do it so regularly. Whenever I talk to them, they say, wait, you guys don't cage your queens? And I think it's just because it is a lot of work, right? You have to find your queen, put her in a cage and all this kind of stuff.
No, it's not. It's much less than put the chemicals in.
But when you're dealing with tens of thousands of colonies, it could add up. But there's so much good evidence, though, that shows how important that is to controlling Varroa mites. So whenever, like you said, is once you cage the queen and you induce that broodless period, all the mice move on to the adults. That makes your organic acids much more effective. So like oxalic acid, whether it's vapor, dribble, whatever, that's gonna be more effective. Formic acid and things like that. What's really good about doing that too is that you could induce that brood break in the summertime. I don't know how it is out there in California, but in the summertime here, probably the next 2 weeks in Louisiana, we go into a hard dearth. And it's too hot, the plants aren't making nectar, there's no flowering plants out there anyway. So by inducing a brood break, it prevents them from getting into this thing of having too many bees in the colonies and not enough food. So that means we don't have to feed them. Makes the treatments more efficacious, especially in the summertime. And then, yeah, you can release that when you get your fall. And like here in Louisiana, we would get a fall nectar flow. So yeah, so caging queens are definitely a part of it. It's just that it's a thing that large beekeepers tend not to do because of the work involved. There's research.
I'm doing this for 5 years already, and it works great.
Oh yeah.
It's not only you don't need to feed bees, the bees not raising next generation of bees and their life expanded. It's like they're living usually when they raise a brood 2 months, and when they don't raise a brood, my bee, I could keep the queen almost for 2 months in a cage, and they live 4-5 months, like collecting honey and don't get sick.
What kind of cages are you using?
I use like they use in Europe, like 12 inches long, 1 inch wide, and how deep the comb is usually, put it in a brood, initially in a brood brood frame.
Mm-hmm.
When they all brood come out, bees fill it with honey, and they never leave the queen alone. They always with her.
Mm-hmm.
Yeah.
So that and so other research that's coming out too is this whole Tropolaelaps mite that's we potentially might be getting. There's some research that just came out from Jeff Williams's lab that shows that that's actually really, really effective for getting rid of Tropolaelaps mites, Varroa mites, all this stuff. So yeah, I think that's something that we'll have to start thinking about implementing, because I think it just promotes general overall colony health with, like I said, not using chemicals. Yeah, for sure. Good one. Yeah.
I was going to ask you about the Propolis. Can you hear me?
Yeah, yeah, I hear you.
I was going to ask about the Propolis mite. Are you doing any genetic sampling of that, or I don't know, just sort of thoughts about other aspects of this kind of research? Is really neat stuff you've done.
Thank you, thank you. Yeah, actually, I'm going to Korea in 2 weeks to do some research on them. So a couple things that we're working on when we're in Korea is I'm going to be doing hygienic testing on the bees in Korea. I've been talking to some Korean scientists and Korean beekeepers, and they were saying they don't do any kind of breeding for any kind of hygienic behavior over there. So I know that the hygienic behavior that we have with VSH bees, is that potentially enough to get rid of the Tropolalaps? I don't know. We'll have to find that out. So we'll be doing some hygienic testing over there, but then also looking at we'll actually be doing combination treatments where we use combinations of oxalic acid, formic acid as our miticide treatment, but then also caging and not caging the queen to see how that will work. Those are the big things that we're working on. We also are working to develop DNA-based methods of detection of Tropolalaps because they're very, very small. So, you know, Varroa is about this big. Tropolalaps might be maybe that big relative to a Varroa mite. So much smaller, harder to detect. And they're also like white, almost clear. So much harder to detect than Varroa mites. So we're using DNA-based methods to do that. We're also bringing back a bunch of samples to do some genetics and genomics on Tropolalaps, looking at viruses and tropy. So I'm taking this big giant liquid nitrogen container with me. I'll know I'll get some looks at the airport, but it's the way science goes. Yeah, it'd be really neat to see if how our VSH bees work with Tropolalaps, because we might have a solution in our pocket. So, but just getting the data to show that is a whole nother bag of worms.
Thank you so much. I'd like to just thank our speaker for this wonderful presentation, just such great information presented so well, and it's also for your horrifying yet beautiful photographs that I have I don't want to say enjoyed, but you know, really like seeing. Thank you for all the education you've given us today. It's really fantastic.
My pleasure, and hopefully this is made available to everybody else who wasn't able to attend tonight. You'll have my email address, send that around. If anybody wants to do some resistance testing, you can send you the kits free of charge. And if you have any questions about Varroa mites, like to hear from you in the future. So thank you very much for inviting me. Have a good night, everybody. Bye.
Good night.